Hello everyone, welcome back to our second lecture video on biochemistry, and we're going to pick up where we left off in discussing polymers. Alright, so let's remind ourselves what a polymer is. A polymer is going to be a large molecule that has been made by linking together many smaller subunits that we call monomers. Alright, over here on the right, if you look, these two smaller molecules here would be the monomers. Connect them together and you get a polymer down here below. Generally, not all the time, but generally we're going to see that as we start to link these monomer units together, that they're going to form a very long chain-like molecule. They'll even appear to be branched at certain times. So an example would be, again, looking over at the diagram, we're going to take two monomer units, both glucose and fructose, two types of sugars. All right, they are monomers of carbohydrates, so here's glucose and here is fructose. Notice how their structures are different, but their formulas are the same. C6H12O6, so they're both isomers of each other. If I link them together, I get a larger molecule called sucrose, which is our regular table sugar that you would put in your coffee, tea, etc. So the idea is, again, we are going to link these smaller monomers together to get polymers. We can build them or break them down based on two distinct types of reactions. So we're going to start with the first type of reaction, which we're going to refer to as dehydration synthesis. So when you hear that word dehydration, you should immediately think about you need water, you're really thirsty, right? All right, you've been sweating profusely, losing water through perspiration, and now you are low in water. Like, that's exactly what it means. All right, so dehydration synthesis is going to be referring to when we take monomer units, we connect them together by removing a molecule of water. So in order to connect two smaller monomers together, you're going to need to remove water as a waste product. All right, what happens is, in order to connect two monomers together, you have to remove an H, or hydrogen group, from one monomer, and an OH, or if you recall, a hydroxide group, from another monomer. When this happens, you destabilize the two smaller monomers, which makes them come together to form a bond to stabilize themselves. So you are destabilizing monomers to make them unhappy. You produce water as a byproduct. And then the two monomers will come together and link together in order to stabilize themselves. And you'll see that momentarily. Again, as a result, for every single bond that you create, so for every two monomers you link together, you will release one water molecule as a waste product. So again, don't forget, each bond or connection will produce one water. So if we just look at it kind of like a simplistic formula, take monomers A and B. All right, you want to connect those together to make polymer C. You're going to take an H away from A, an OH away from B. You produce water as a byproduct. And when you do that, you destabilize A and B so they come together immediately to try to make themselves happy again to form the larger molecule of C. So if we look at a visual here, I have a short polymer right now that is made up of 1, 2, 3 monomer units, and it's growing. I want to make it even bigger. Notice that any time that you see either a monomer or a polymer, there's going to be an OH group on one end and an H on the other. So if I want to add another monomer unit, if I want to add a fourth, what I need to do is remove an OH from this monomer, an H from this growing polymer here that produces water as a byproduct, and now the fourth monomer will link together to this growing polymer chain, and your polymer is even longer. All right, so again, the idea is you're removing water to add a monomer, and for every bond that you create, you're producing one water molecule as a waste product. So down here in this picture, for this polymer that is now 1, 2, 3, 4 monomers long, how many water molecules would have been produced as a waste product? Answer is going to be, look at the bonds between the monomers. So right here would have been 1 water molecule. Right here would have been 2. And right here would have been 3, for 3 total water molecules to create a polymer chain that is 4 monomer units long. Now that's building. What if I want to break down a polymer back into its individual monomer units? Well, that reaction is going to be called a hydrolysis reaction. All right, hydro means water, right? Hydro, I'm going to need water this time. So hydrolysis reactions refer to the breaking apart of a polymer, back into its individual monomer units. This is pretty much what we call digestion, right? So hopefully you've had some breakfast this morning, lunch, dinner, whatever time of day it is for you. And digestion is going to be hydrolysis reactions, breaking down larger food molecules into their individual monomer units. This time, you're going to need water as a reactant in your formula. So this time, it's the reverse for a hydrolysis reaction you can see down here in the formula. Now I'm going to take a polymer. I'm going to require water in order to break it down back into its individual monomer units. All right, both A and B here. So what you do with the water is you split the water back into its two ion forms, H and OH, or hydrogen and hydroxide. So once you break the bond between monomers, the H and the OH can go right back into place and stabilize the A and B that they had originally been removed from. So if we look at a picture of this one, up top we have a polymer right now that is made up of 1, 2, 3 monomer units. If I want to break it down into all of its monomer units, I'm going to need 1, 2 water molecules in order to separate all of them. So that's why you see 2 water molecules coming in. So imagine right here where my cursor is flying around. What you're going to do is you're going to break that bond between those two monomers. So imagine then you have something like this. All right, and what we're going to be missing here is the oxygen on the one, but now that oxygen is missing an electron, as well as over here where I'm circling, there's nothing there to match the electrons that were needed originally. So here comes water to the rescue. All right, what you would do is literally take the water, split it into H and OH. The hydrogen ion I can put right here onto that oxygen to stabilize it. And on the other monomer unit, I can put an OH group to stabilize that. So now both ends are going to be perfect and stable. And you can see down below, I now have 1, 2, 3 individual monomer units that are ready to go. And they've been completely broken down into those single units. Now let's see this in action. All right, if I get out of this momentarily and come over to this animation right here, let's look at the process of digestion here a little bit more closely. So again, here's our organ system in your belly and your chest. Here's your liver, here's your stomach, here's your large intestine, small intestine on its way out the other end. Let's see how this plays out.
An example of how enzymes function in the body is from the enzyme sucrase. Sucrase resides on the surface of the microvilli on the intestinal epithelial mucosal.
So you have all these little kind of like fingers or hair-like projections that are inside of your intestines. These are what are grabbing nutrients from the food that you're digesting.
Cell surfaces. This animation presents a graphical representation of the way that sucrase catalyzes the hydrolysis of the common disaccharide sucrose, which we know as table sugar.
So here's an enzyme. Enzymes are actually proteins. They help us with these types of reactions, both dehydration and hydrolysis. So here's sucrose, the ball and stick model. All right, this enzyme is going to help us break this down into 2 individual monomer units, and then also water will come in here to the rescue.
Into its component monosaccharides, glucose or blood sugar, and fructose or fruit sugar. Hydrolysis is accomplished because when the sucrose molecule binds to the active site of the enzyme's configuration is changed so that the oxygen bridge between the 2 monosaccharides is exposed to water molecules in the solvent.
So this enzyme we'll learn about later this unit is helping us out, making sure that this little bridge here, that oxygen connecting the 2 monomers, is exposed to allow water to come in and break it.
This exposure permits a water molecule to actually break the bond, the oxygen bridge, and attach the components of water, an OH to one of the monosaccharides and an H to the oxygen, which is still attached to the second monosaccharide. This effectively cleaves the bond between the two monosaccharides and converts the disaccharide.
So you can see now that an H from the water is on one end and an OH from the water is on the other end, stabilizing now these two individual monomer units
Into two separate sugars. Once this is accomplished, the enzyme's configuration is changed back to the original shape. The two monosaccharides float away, and the site becomes available for another sucrose molecule to bind, change the enzyme's configuration, and be hydrolyzed. This action can be repeated many times until the enzyme becomes denatured, is inhibited, or just wears out.
All right, so that is a hydrolysis reaction that's going on right now live in your body, especially if you had some breakfast. So I bring this back up. In conclusion, again, we have two types of reactions that we're going to use to both build and break down polymers. All right, if we want to build polymers, we're going to use what's called a dehydration synthesis reaction, where you take two monomers or more, and you're going to link them together by removing a water molecule. So you can see here again where my cursor is. I need to destabilize these two monomers. To do so, I take an OH off one and an H off the other. There's where my water comes from. And now here's that oxygen bridge that we also saw in the animation to connect and stabilize these two monomer units. Or in reverse, let's say now you want to break down that polymer that you made. Maybe you made this to store energy for long term in your body, but now you need that energy, so you want to start to break it down. So it just works in reverse. All right, for every bond that you create, you produce a water, but now for every bond that you break, you're going to need a water molecule to separate it. So again, if I break this oxygen bridge right here, the two monomer units will be unstable. So here comes water to the rescue. An OH is plugged back in on one, and H on the other to stabilize it. Both smaller monomer units are now happy, and I could use them for whatever I want. So let's say if this was just a protein that I broke down, and I have now separated it into its individual amino acids, maybe I can now use these amino acids to build some muscle, especially if you just got done working out. All right, if you have any questions, make sure that you reach out to Ask Me as soon as possible. Thank you.